Warning: the SCP Foundation database is classified. Access by unauthorized personnel is strictly prohibited. Perpetrators will be tracked, located, and detained.
Item Number: SCP-047. Object Class: Cater. Special Containment Procedures: SCP-047 is to be contained in a 0.5 meter by 0.5 meter by 1 meter hermetically sealed storage box at all times. This box is to be locked in Storage Locker 047-A inside P3 Secure Biohazard Lab 047-B. Any entrance to and activity inside 047-B will be recorded by biometric scan, closed-circuit camera, and
Redacted.
Entry to 047-B requires the authorization of the project manager, in addition to at least one O5 level clearance. SCP-047 is to be treated as a Priority 4 contagious biohazard in all protocols, including mandatory quarantine if exposed. Suite Q-047, adjunct to Lab 047-B, has been provided for this purpose. In the event of outside contamination of SCP-047-1, Lockdown Protocol 047-01 Yersinia must be engaged. Description: SCP-047 is a heavily rusted, breached gas cylinder made of an iron
Redacted.
alloy. When exposed to open air, the material of the cylinder sublimates slowly, producing a previously undocumented mutagenic gas. This gas has no effect on eukaryotic organisms, such as humans, but profoundly alters prokaryotes, showing preference for common human microbiota, the natural microorganisms that live on the skin and throughout the body. On rare occasions, these mutations produce a superbug, collectively known as SCP-047-1, a natural commensal with enhanced survivability and therefore opportunistic pathogenicity. The pattern of changes induced by SCP-047 suggests that these highly infectious microbes are, at least to some degree, selected for. Although the specifics of SCP-047-1 species are dependent on the base bacterium from which it is derived, there are several characteristics which appear to be generally consistent across all cases of SCP-047-1 mutation. Enhanced survivability in the bacterium's natural environment and similar environments, full-spectrum antibiotic resistance, increased reproduction rate and consumption of available material, development of a sporulation ability in gram-positive bacteria, increased ability to uptake, hold, and share plasmids, particularly in gram-negative bacteria, and increased transmission due to traits described above. SCP-047-1 samples are normally debilitating and virulent. However, compared to other Cater-class SCPs, it should be noted that SCP-047-1 have a relatively low mortality rate due to their action through mundane biological pathways. Several strains of bacteria have been selectively mutated by SCP-047. Mutation of bacteria in culture is possible, but the process appears to be much more effective with bacteria living on a human host. Generally, mutation of natural commensals for experimental purposes is encouraged. After the containment breach of January 21, 2019, see Incident Report Yersinia 047-01, 2019, mutation of already pathogenic species is banned and all existing samples must be destroyed. 3 particular species of SCP-047-1 mutated bacteria are of note due to their involvement in the containment breach of January 21, 2019. Propionibacterium 047A, or P047A, is a strain of Propionibacterium acnes mutated by SCP-047. Pathogenicity: severe skin colonization around sebaceous glands. Modification of skin pH to levels that become toxic to skin cells. Massive inflammation and immune cell infiltration. Eventual breakdown of skin structure leading to sepsis. Transmission: transmitted by skin-to-skin contact. can remain active on inorganic surfaces for up to 5 hours. Lethality, approximately 40% mortality rate, runs its course in 2 to 6 weeks. Very visible symptoms within 5 to 10 hours. Contagious within 2 to 5 hours. Handling. As soon as visible symptoms form, victims must be quarantined. Deceased victims should be incinerated. Streptococcus 047C, or S047C, is a strain of Streptococcus mitis mutated by SCP-047. Pathogenicity. Causes inflammation of the mouth and esophagus initially. Leads to open sores in the mouth, which result in S047C entering the bloodstream and becoming septic. Death is usually due to infectious endocarditis. Transmission. Droplet. Can remain active indefinitely by sporulation. Lethality. Approximately 35% mortality rate. May become a recurring chronic condition if non-lethal. Handling. Subjects with any sign of mouth infection should be quarantined. Deceased victims should be incinerated. Clostridium 047A, or C047A, is a strain of Clostridium difficile mutated by SCP-047. Pathogenicity. Unknown. C047A was developed from tissue culture and has never been exposed to a human. No samples remain in Foundation control. Transmission. Unknown. Presumably transmitted through fecal contamination, as with Clostridium difficile. Due to smaller, more robust spores, may also aerosolize with flatus. Effects of aerosol intake of C047A cannot be predicted. Lethality. Unknown. Presumed extremely high risk of destruction of endothelial lining of gastrointestinal tract, leading to inflammation, sepsis, and toxic megacolon. Handling. Until further research has been done, victims should be quarantined and placed under 24-hour medical observation to develop functional diagnostics for this strain. Deceased victims should not be incinerated until adequate etiological research has been performed. Recovery log. SCP-047 was recovered from the site
Redacted.
Secure Laboratory by a Foundation biohazard recovery team in response to a full compromise situation on
Date redacted.
1990.
Redacted.
Testing logs indicate that the research team was attempting to contain
Data expunged.
in a Class
Redacted.
SCP-stable pressure cylinder, which led to
Redacted.
combining with
Redacted.
a full molecular biological analysis of this is available in
Redacted.
the initial release of gas when SCP-047 was structurally compromised was sufficient to cause a microbial bloom of uncounted species of SCP-047-1, killing all staff in the lab within
Redacted.
hours. Exposed site
Redacted.
staff obeyed standard Foundation quarantine and containment protocol, and the infection was contained successfully. Incident Report Yersinia 047-01, 2019. SCP involved. SCP-047. Description: on January 21, 2019, at approximately 0300 hours, Storage Locker 047C, Site
Redacted.
Lab 047B, containing bacterial samples mutated by SCP-047, was compromised after a complete simultaneous
Data expunged.
leading to failure of security measures in the area. 3 samples, of a total 12, were stolen.
Data expunged.
Since the initial containment break, outbreaks of one of these stolen bacterial strains, Propionibacterium 047A, or P047A, were recorded globally in communities of increasing size and population density. Further information on stolen material, spread, and containment follows. Compromised items: Propionibacterium 047A, Streptococcus 047C, and Clostridium 047A. See SCP-047 for further information. Outbreak information. First outbreak, P-047A, February 18, 2019, Siberia. Contained. See Incident Report P-047A-02-2019. Second outbreak, P-047A, March 21, 2019, Northwest Territories, Canada. Contained. See Incident Report P-047A-03-2019. Third outbreak, P-047A, April 18, 2019, South Australia. Contained. See Incident Report P-047A-04-2019. Fourth outbreak, P-047A, May 18, 2019, Mato Grosso, Brazil. Believed contained. See Incident Report P-047A-05-2019. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P-047A and be on the lookout for possible infection. Fifth outbreak, P-047A, June 17, 2019.
Redacted.
Iraq. Site immediately.
Data expunged.
Which is believed to have contained the infection. Access to Incident Report denied without O5 clearance. Sixth outbreak, P-047A, July 14, 2019, Cameroon. Quarantine enacted. Efforts to track outgoing civilians underway. Infection not contained. See Incident Report P-047A-07-2019. Seventh outbreak, P-047A, August 15, 2019, Dalarna, Sweden. Quarantine enacted. Believed contained. See Incident Report P-047A-08-2019. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P-047A and be on the lookout for possible infection. Eighth outbreak. Not recorded. Believed to have taken place in North Korea.
Data expunged.
Agents with government access are attempting to gain access to parallel information, but due to
data expunged,
local services have been extremely uncooperative. Containment status unknown. Ninth outbreak, P-047A, October 11, 2019, South Carolina, United States of America. Quarantine enacted. Efforts to track outgoing civilians primarily successful. One civilian in a pickup truck is believed to have
data expunged.
Infection not contained. Resolution. Reports from
data expunged
indicate no further outbreaks are believed likely, but agents are advised to be on the lookout for new flare-ups resulting from uncontained civilians in previous outbreak regions. These may continue for years to come due to P-047A sporulation. Investigation into the cause of the initial compromise is underway. Anyone with useful information may anonymously contact security.